Well, good morning, good evening, good afternoon, wherever you are. Thank you very much for joining us today for the latest in our series of ACT-Accelerator media roundtables. I'm Charlotte Baker, the communications lead for the ACT-Accelerator. So today at this roundtable, we're going to be focusing on R&D, so the latest research and development in COVID-19 tests, treatments and vaccines. And I'm really delighted to be able to welcome an absolutely stellar panel of experts from 4 of our ACT-Accelerator partner agencies who are going to be giving you a rundown today on where we are in the latest developments with regards to COVID-19 tools, and also crucially underlining the role of R&D still, both as part of ACT-A's work and also in a wider sense. So our speakers today, just to remind you, are WHO Chief Scientist, Dr. Soumya Swaminathan, UNITAID Director of Strategy, Janet Ginnard, CEPI Executive Director of Research and Development, Dr. Melanie Saville, and FINE Chief Technology Officer, Marta Fernandez-Suarez. So a few things to bear in mind: this briefing is on the record and will be focused exclusively on research and development. So please keep your questions confined to these topics. Speakers will present for 5 minutes each at the beginning, and then we will have a Q&A session. So without further ado, I think we'll get going. I'd like to hand you over to WHO Chief Scientist, Dr. Soumya Swaminathan, who will start us off today. Soumya, over to you.
Thank you, Charlotte. Hello, morning, everyone. So where are we today in the pandemic? Actually, we're at a quite a challenging phase. I think it's also a time of some uncertainty. We see that finally, after several weeks of very high case numbers and high deaths, we've gone back to about 70,000 deaths a week being reported over the last several weeks. Last week, for the first time, we've seen a decline in almost all regions of the world, both in terms of cases and in terms of deaths. But we're still at 60,000 deaths a week. So we still are not even back down to what we were in before October of last year. The decline in cases also is partly linked to the countries, many countries having taken the policy of less testing. So we can't obviously go just by the numbers, but I think the deaths, particularly we are seeing in some countries in the Western Pacific region now, explosive increase in cases as well as increase in deaths. And when we analyze a country like Hong Kong, for example, and see why their ICUs are full and why their mortality rates are so high, especially among the elderly, we find that it's actually related to vaccination coverage. And in the above 80-year-olds in Hong Kong, vaccine coverage is 30%, and it's about 50% in the 60 to 80 age group, and it's actually higher in the younger age groups. And so this is it goes to show that we're not out of this pandemic. That's number one message. We still have 60,000 people dying a year. Secondly, that across the world, high-income, middle, low-income countries, you have challenges in vaccination coverage, and we can talk more about that. And thirdly, the variants, particularly Omicron, and now the sub-lineage BA.1.2, which seems to be overtaking BA.1 in many countries, shows that this virus is continuing to replicate, it's continuing to evolve, to acquire mutations. We've also seen reports recently of animal that are infected. We had the white-tailed deer in the US. There's another species of deer that's been reported. And then we have, more than a dozen species of animals that we know can be infected. So there's also the possibility of recombinants developing. So future scenarios can be broadly divided into one, that this settles into an endemic disease, which means you have a constant background rate of transmission. You'll have also deaths happening always, because there'll always be vulnerable people, those who are immunosuppressed, those who have not been vaccinated or who've had waning immunity. So we will see that. And then you may have periodically outbreaks, could be seasonal or could be otherwise. The second scenario is that we will get another variant, and that variant is likely to be more transmissible than Omicron, because any variant to be successful in transmission has to be more transmissible than the previous variant. But whether it will also have mutations that make it cause more severe clinical disease, we don't know. So you may get a variant which causes milder disease, or a variant that causes more severe disease, and we need to be prepared for that scenario. It is not impossible. It could happen. And then, of course, we could have a scenario where you have the thing completely, recombining in animal species, and that's kind of the worst case scenario, is that you have almost a new virus emerging. Meanwhile, of course, the threat of pandemics due to other viral families has not gone away. And so we have to keep in mind that anytime you could have an influenza pandemic or some other viral pathogen arising, again, most likely zoonotic. So in addition to focusing on COVID, we need to build our R&D capacities to be able to address all of these other, and that's why the CEPI portfolio, for example, on many of the neglected diseases is very important. I'd like to also mention that the mRNA manufacturing hub and spokes that we're setting up now, again, has a goal which is longer term and beyond COVID. So the idea really is to build a network of labs and of manufacturing sites, mainly in LMICs, that develop the competency to make mRNA-based products. Could be vaccines, could also be biologics. So we have two parallel streams. One where the mRNA technology that is either transferred to us from a company that has a technology or that's indigenously made by the scientists at the hub in South Africa or in one of the other centers is then licensed out and shared with others. The potential here is huge, because if you have distributed manufacturing sites that are developed over the next couple of years, and many governments are now investing in those facilities, in case of a future pandemic, you will have the ability to quickly, as soon as the genome sequence is known, be able to make mRNA vaccines around the world. And even if those sites are relatively small, you can still produce hundreds of millions of doses fairly quickly. So that's one advantage. The other is, of course, that we're training a workforce. So we have the biomanufacturing workforce training program and hub that was announced. And so all of these countries that many of whom don't have capacity will now be able to send people to South Korea for training in basic biomanufacturing, and then, of course, more advanced training depending on the new technologies. I think we still have a long way to go in terms of second generation vaccines, which can block infection. We saw that the vaccines did well against Omicron for severe disease, but not against getting the infection. And so we still need to invest in mucosal vaccines, second generation vaccines, more scalable, that are less costly, that are room temperature or 4 to 8 degrees stability, and that provide mucosal immunity. So investment in vaccine research needs to continue. Clinical trials need to get done. Investments in therapeutics also need to continue. We don't have a lot of therapeutics, though we do have at least 2 drugs now and than we had before. And we also need to, of course, continue diagnostics. And again, we can go back and look at the figures, but we have such huge inequities in access to diagnostics. So the whole test and treat strategy is going to need investments in that. And then finally, investments in sequencing capacity, which again, we're working with a network around the world that started with influenza. Labs, but have now built their capacity, and more labs are getting developed. But we need a global data sharing system and a platform. GISAID has worked well for us so far, but GISAID has limited to influenza, SARS-CoV-2, and RSV. So we need these global data sharing platforms and principles and governance of such systems so that we can have the rapid and timely data sharing, which should not be accompanied by punitive action, as was seen in South Africa, when a lot of countries shut their doors in South Africa because they reported Omicron. So this is why you need a global treaty or an understanding or a convention on how one should treat these data and how one responds to it without putting any countries at a disadvantage. So I'll stop there, and we can come back later to more questions. Back to you, Charlotte.
Thank you very much, Dr. Swaminathan. I'd now like to invite Unitaid's Director of Strategy, Janet Ginnard, to take the floor. Over to you, Janet.
Thanks very much, Sumia, and also to Charlotte for the introduction. As mentioned, I'm Janet Ginnard, Director of Strategy at Unitaid. I'm speaking on therapeutics today. We use therapeutics to refer both to medicines and to medical oxygen. I'll focus on medicines today, given the emphasis on research and development. Certainly appreciate the comprehensive scope today because there is no single solution to COVID. We need vaccines and treatments and the tests to guide their use. With the situation that Dr. Swaminathan described, we can expect to see continued transmission, cases, deaths, new variants, and we'll have a continued need for medicines. So at Unitaid and partners in the ACT-A therapeutics pillar are working to secure equitable global access to effective medicines. We focus on end-to-end support, leveraging the expertise of the different partners within the ACT-A collaboration. And we do that by addressing research gaps, closely tracking products that are emerging from the pipeline or advancing through the pipeline to identify work that can be done in advance to enhance supply security, access. We devise implementation strategies, including test and treat for some of the orals. We'll come back to that in a moment, and deploying critical tools at scale. A couple of our successes to date, Sumya had mentioned the living guidelines from WHO that are critical for many countries to adopt new products. The guidance includes positive recommendations for several therapeutic options, including work in progress, guidance in progress for the very first outpatient oral antiviral therapeutics. So the first oral pills, guidance on molnupiravir just today, in fact. There's voluntary license agreements with industry partners, MSD and Pfizer so far, that have been enacted through the medicines patent pool that enable a robust generic supply base to be put in place for serving about half the world's population. Unitaid, together with FIND, are driving early introduction, informing test and treat strategies in 22 countries in Africa, Southeast Asia, Latin America, and the Western Pacific. And Global Fund and UNICEF are working together with the Emergencies Program at WHO, organizing initial supply and procurement of therapeutics. So we've been working very closely with WHO throughout this, tracking the pipeline to inform all of this work in advance, at risk when necessary, and continuing to identify research and development support that may be needed. So in addition to WHO, Wellcome, the Gates Foundation, Unitaid are supporting platform trials, expansions, increasing the scale and the global reach of evidence generation. That's including the Antikov trial in low middle income countries, the extension of the recovery trial to Southeast Asia and Africa. There's a need not only for evidence on the new products to secure initial registration, but also on how they can best be used, also in combination therapies. For the year ahead, we have a focus on oral antivirals for outpatient treatment. As Sumya had mentioned, to date, the cupboard has been a little bare and not as many treatments as we would like to see, but we're at a very exciting time. We're on the cusp of a paradigm change really in treatment with products that are oral, can be used for outpatient treatment in high-risk populations, in high-risk people to dramatically cut the risk of hospitalization and disease progression. So I'm talking about products like MSD's molnupiravir, WHO recommendations published just today, Pfizer's nirmatrelvir ritonavir under review, fluvoxamine, that's an older product for other indications, but some evidence of activity in COVID. Shionogi is a company in Japan with a pipeline antiviral, just having announced promising results from phase 2 trials and filing for emergency use approval in Japan. So many of these are exciting because many of these can offer a widespread, scalable way to stop disease progression, reduce hospitalization, ease the strain on healthcare systems, and stem new variants. So this complements continued efforts on the established or already recommended drugs, many of those for severe and critical COVID, WHO working hard to review the evidence on the newer variants and the efficacy or changes in efficacy that we might see. It also complements work on oxygen, again, a mainstay of life-saving inpatient treatment. So the key challenges that we're working to address, just to summarize, equitable access to products. Ideally, there's a diversified generic supply base that's established and available for rapid deployment and scale up as soon as WHO recommends a product. We may need to look at originator stock to bridge to availability of supply from other sources, generic sources. So ACT-A partners are working with companies like MSD, Pfizer, Roche, others, and engaging with companies that have pipeline products that could play a global role. Shionogi I mentioned. So adequate, affordable stock of products when recommended are really essential until generic options or other alternatives can be sourced. We're also focusing on the challenge of country readiness, country engagement, intensified efforts to engage with countries in devising scalable test and treat strategies, strengthening oxygen systems, securing affordable supplies. The linkage to tests is something that we'll come to in just a moment. And then finally, financing. $3.5 billion needed for ACT-A therapeutics, and most urgently, over $1 billion for procurement, advanced procurement of oral antivirals for high-risk patients and for procurement at scale. So thank you for your attention and looking forward to the Q&A and discussion. I will hand over to Melanie Saville, CEPI Executive Director of Research and Development. Thank you.
Thank you, Janet. And I'll start off with giving you sort of an overall of the current status of vaccine R&D. And there are more than 300 vaccines in development. And to date, there are 9 vaccines that have received the WHO Emergency Use Listing, EUL, of which 3 of those have been supported by CEPI. And we have seen that vaccines have been able to break the link between infection and severe disease and death, where those vaccines have been available. But we must also recognize, as Sumia has already mentioned, that we need to continue to work on R&D for vaccines, looking at next generation vaccines, and some of the areas have already been highlighted, like looking at transmission blocking vaccines, looking at improvements to vaccines in terms of cold chain, in terms of cost of goods, to make vaccines more affordable and more accessible. I think it's important to mention a platform that is now becoming licensed and EUL'd. This is the protein-based platform. There are a number of protein-based vaccines that are licensed and widely used globally. CEPI specifically had funded 3 advanced candidates: Novavax, which has now been received EUL, Clover, and SK Bio. Together with another advanced vaccine we should mention is the Sanofi GSK protein vaccine, which is very advanced in development and has good results recently, are on the pathway there to licensure. And they have, through the work that they've done with other vaccines for other infectious diseases, been shown to have good thermostability, a favorable safety profile, generally low cost, and the vaccines that we have seen with data for COVID-19 have generally shown a good broad immune response, and with the adjuvants, potential help for vulnerable groups. I think it's important to specifically mention the R&D towards vaccines to look at variants. We have seen, obviously, as new variants of concern have arisen, that there is always the concern of what the impact of those variants might have on the effectiveness of the vaccines. And there are several approaches that are being taken in terms of R&D to really look at how to better tackle variants in terms of vaccines. And there are really 4 main areas, so I'll just briefly mention those 4 areas. The first is really looking at optimizing the use of the vaccines that we currently have. So looking at what are the best regimens, whether that's a homologous or heterologous regimen, when are boosters needed, and how can that help give you a more broad immune response. CEPI is specifically funding clinical trials to generate evidence on some of these questions with a focus on low and middle income countries. The second area is looking at developing vaccines against the variants as they emerge. So this is in the context of an existing licensed vaccine and changing the strain very much as you do with flu. And we have seen, for example, with Moderna and Pfizer-BioNTech, that they are advancing their Omicron variant vaccine into the clinic very rapidly. CEPI is also funding some early work on variant approaches to vaccines, such as the Omicron variant for the Gritstone bio project funded by CEPI. However, this step, when we're looking at variants and developing vaccines against variants as they emerge, we still remain one step behind the virus. And what we want to do is get one step ahead of the virus. And so there is a concerted effort at looking at broadly protective SARS-CoV-2 vaccines, if you like, variant-proof vaccines, where using innovations in antigen design, creating vaccines that will protect broadly against the variants that exist and potentially the variants that will continue to emerge. And then taking that a step further, really looking at universal coronavirus vaccines. SARS-CoV-2 is the third coronavirus outbreak or pandemic in the 21st century. Efforts are being put in terms of R&D towards looking at a broadly protective vaccine that could prevent not just against SARS-CoV-2, against SARS, MERS, and future potential coronavirus outbreaks. And to date, CEPI has invested in $90 million US dollars for development of 6 different approaches to either broadly protective SARS-CoV-2 or a more broadly protective coronavirus vaccine. So with all of this in mind, as well as looking at tackling this pandemic, we need to take everything that we have learned from this pandemic to really start preparing for future pandemics that we know will hit. So continuing to really develop the vaccines. We know that in SARS-CoV-2, we got to the first vaccine use in 326 days. We set the challenge of what if that could be done in 100 days. And there are a number of things that can be done, and we have actually outlined some of those through really broad consultation with the vaccine R&D community on looking at how can we get to 100 days through a perspective piece in the New England Journal that was published yesterday. G7 and G20 have also taken on the 100-day ambition for vaccines and also applying that to diagnostics and therapeutics as well. CEPI is holding a global pandemic preparedness summit next week with the UK government, where we will be looking at preparing for pandemics in the future and raising funds for the CEPI's next 5-year plan of $3.5 billion US dollars. So thank you very much. And I think now I can hand over to Marta Fernandez Suarez, who is the Chief Technical Officer at FIND.
Thank you very much, Mel. And thank you everybody for being here today. So I have a few slides that I'm just going to be narrating very quickly. So next, please. I think we all know that in order to tackle the pandemic in the past 2 years and moving forward, we need an arsenal of diagnostic tests that are aiding us in clinical management, vaccine efficacy, identifying vaccine efficacy and seroprevalence, as well as in surveillance. And that we need tools that are available on the different levels of the healthcare system, all the way from centralized hospitals and laboratories to decentralized centers in primary healthcare and beyond. And as we've seen in the COVID pandemic has shown us what can be done by empowering patients with their own home tests. We have this arsenal of tools, but still today there is a trade-off. Generally, tools that are used in centralized settings have improved performance, whereas as we move into decentralized settings, the performance is not as good, perhaps still clinically relevant, right? And that depends on the use case and the intended use of the technology. And then the cost also become a barrier. So there is still a lot of R&D that has happened over the last 2 years, and that still need to happen so we can bridge this gap between performance and accessibility on diagnostic tests. Next, please. So this is just a graphical way of showing, we are doing, when the pandemic first started, the first tests we were able to see were PCR tests, molecular tests that are done in the lab in these big machines, very high throughput. And those are great because They are much faster to develop. They are faster to start implementing and give us a handle on the pandemic and on the number of cases. As the pandemic moves forward, we started developing tests that allow us to bring testing to a more decentralized settings, like in the primary care. And these are just pictures that highlight some of the platforms that we have. So these are integrated molecular tests that can now be done at primary healthcare. They can be done in an emergency room and in others, as well as the rapid tests that now everybody is very aware of. Next, please. The pandemic evolved, and what we saw was this huge shift to self-testing, right? And I should say, just like Sumia was highlighting before, there is still a huge equity gap in between what is available in low and middle income countries and in high income countries. So in this next slide, I'm just showing you where we are today. There has been this explosion of tests, right? So we went from a few tests in the lab, a few at the primary healthcare, to through huge amounts of investments, both private investments, also investments through the ACT-Accelerator, investments from the US government, the European government. We've seen now have a suite of tools that can be used at the different levels of the healthcare system, right? And we go all the way to the left. We have now seen that for the first time, we have now regulatory approved tests, and those are on the lower left, or the upper left corner, regulatory approved tests to be used at home that are molecular tests. So relatively high performance tests that anybody can take in their house. Some of these are also connected, so they allow us to report testing. We are seeing a huge amount of self-tests that have been adapted both on the antigen front. And then in the middle, you can see that we went from like a single platform for molecular testing at primary care or decentralized settings like emergency rooms and others, to a whole suite of platforms from different companies. We're not done yet. A lot of these tests are still very expensive, and there is still a lot of work that needs to be done in making them more robust. And for example, I think this is a theme that has come across in therapeutics, vaccines, and now diagnostics, stability. A lot of these tests have been developed in regions where there is cold chain, where tests can be shipped with ice or others. If we are to improve access to this new suite of tests in broader regions of the world, we need to work on stability, and we need to work on cost, so we can now bridge accessibility, performance, cost, and everything that involves. Next, please. Just a few words on digital, right? Because I think what I've been describing so far is everything that is happening and we're still investing on for hardware, for more tangible devices. But we've seen an explosion over the last 2 years on digital tools that connect these diagnostics to the healthcare systems and that enable task shifting, right? Is something that we at FINE and other ACT-Accelerator partners are putting a lot of emphasis. How can we enable lower levels of the healthcare system all the way to the patient to use diagnostic tests that were traditionally restricted to laboratory and healthcare professionals? So we've seen obviously a lot of apps. You may have used some of them for reading rapid diagnostic tests. We are seeing a lot of apps that are used for guiding the patients on their own symptom screeners. We need to go further, right? We need to make sure that a lot of these home-based use tests become useful in the next phases of the pandemic, which may involve differential diagnosis of COVID versus other diseases or respiratory diseases like flu and RSV and others. But when it comes to symptom screenings, to really making them general, so the patient, the individual at home can also be guided through those different symptoms. We also have new diagnostic tools to enable the healthcare workers, such as the use of artificial intelligence and machine learning to interpret X-rays. We pioneered that on the use for tuberculosis in close collaboration with WHO, and now it's not also being applicable to COVID. There is still a lot of improvements that need to be done on those algorithms to make them broadly applicable to a wider set of populations and for other diseases. And also just a quick touch base on the digital tools for surveillance, right? And I think everybody knows there has been an explosion on all of these tools, all the way from privacy preserving proximity tracing apps to mobility pattern analysis for predicting hotspots and others. A lot of active partners are now working on surveillance tools and on how to address the gaps of aggregating all of the surveillance data that comes from epidemiological and genomics at both a national and a global level to inform decision making. Next, please. So I don't need to touch much on sequencing, as Sumia already mentioned it in her opening remarks, but sequencing has also exploded over the last 2 years. A lot of the R&D innovations that we've seen have come into bundling those products. We used to have different products that enable the whole sequencing cascade, right? It goes all the way from collecting the sample to preparing that sample, preparing the reagents, then running the sequencer, and then doing all of the bioinformatic analysis. Now, the companies are now coming with those bundles, and that has been proven tremendously helpful for adoption. And we now are seeing about the end of the December last year, we have seen that 7 million SARS-CoV genomes have been uploaded to GISET, as Sumia mentioned, from 220 countries. And we have seen a reduction in the time that it takes from sample collection to having public access, right? It was only about 48 hours for the Omicron surge. So really, a lot of what we're doing now is on focusing, increasing focused capacity building for sequencing, and on the R&D front, developing these workflows that made countries more efficient at the use of their resources. Next, please. So just finishing here to say that there's been great innovation. Some gaps still exist, as I mentioned, but the biggest gap today is with all of these new tools that we have available, how do we make sure that there is equitable access to all of these tests so that we can stimulate the rapid and the effective uptake in country. Next, please. So just 2 more thoughts, and this is something that has been also mentioned on the vaccine front. Some of the initiatives that FINE and other partners together with Unitate and others in the ACT-Accelerator, we are investing on technology transfer to facilitate regional manufacturing. This is for disruption, to tackle the problems with disruption in supply chain issues, but also to really build a sustainable system for today and moving forward. And then next, please. In addition to rocket manufacturing, one of the other activities that we're working on, and this is also in collaboration with Unitate and other active partners, is exploring different service delivery models beyond the traditional healthcare settings. What is the impact of doing testing in workplaces and in the schools, at the homes, through pharmacies, at border crossings, and others? So I think I'd leave it here, and with this, I just hand it over to Charlotte, who's going to open the floor for questions.
Thank you so much, Marta, and thank you to all of our speakers for those, I think, really interesting and thorough roundups of where we are on R&D with regards to therapeutics, vaccines, and diagnostics. So Anne's already ahead of me, but I was going to say to any of the reporters with us, if you'd now like to ask questions, then please put your hand up or drop a question into the chat, and we will get going without further ado on the Q&A. So just a reminder that when you ask your question, ideally, please say who it's directed to as well. That helps us a lot. So Anne, would you like to unmute yourself and ask your question?
Hi, thank you, Charlotte. You can hear me? Okay, thanks. I have one question for Janet Ginnard and 2 for Mel Saville. Is that okay that I just, yeah, okay. So the question for Janet Ginnard, what is the latest on the negotiations between Unitate and Pfizer on supply of originator Paxlovid to the ACT-Accelerator over the coming months while we're waiting for the generics to come online? Does it look like Pfizer is going to supply any Paxlovid through the ACT-Accelerator over this year? And then 2 questions for Seppi, please, for Mel. The first one speaks to companies' adherence to the equitable access provisions of their CEPI deals. Novavax got almost $400 million from CEPI in 2020. And after some delay, there have been recent reports that the Novavax vaccine is now being supplied to various countries in Europe. So my question is, have deliveries of Novavax via COVAX begun? And if not, when do you expect that to happen? So that was my first question. And then the other question is about the 100-day mission. I've heard it argued that the 100 days can only be achieved with mRNA vaccines, and that, notwithstanding the new mRNA hub, that there's a danger that this will therefore only be relevant for high-income countries. And so if you could please comment on that. I hope those questions are clear. Thank you.
Janet, do you want to take the first one and then we'll go to Mel?
Sure, I'm happy to. Thank you. So just on the latest on negotiations with Pfizer on originator product or access to originator nirmatrelvir or ritonavir. In fact, ACT-A procurement partners, Global Fund and UNICEF are negotiating directly with Pfizer. Those are ongoing negotiations, so I can't comment on specifics, but more generally, we've been calling for actions to ensure that all low middle income countries have early, equitable, affordable, and flexible access. We'd love to see expansion of the scope of the geographic scope of the list of countries that are eligible for licensing through the Medicines Patent Pool, a low transparent price for ACT-A partners that's applicable to all low middle income countries, and any other actions that could help accelerate generic manufacturing. But I'm not in a position to comment on the specific negotiations. They're ongoing. They're Global Fund and UNICEF. Thank you.
Thanks. And to answer your two questions, so first of all, on the equitable access provisions. So indeed, in terms of CEPI funding, equitable access is at the core of what we do. And we do have our access provisions for the vaccines that we have been funding and make those available, or first rights to vaccines for the vaccine that we are developing. So vaccines, Novavax specifically, indeed, is being made available also obviously through the APA that is with Gavi as part of the COVAX facility. And the doses that are being made available are going through the usual allocation system through Gavi, through WHO, through the fair allocation system. To your second question on the 100-day mission, I think it's fair to say that there is a strong emphasis on mRNA as a rapid response platform that could meet the 100 days. It is not the only mechanism, and we have to say that not every vaccine will be best fit on mRNA necessarily. It is important to further develop the mRNA technology, both to develop other vaccines, prototype vaccines, but also to look at some of the areas that you're mentioning, like low cost of goods, better thermostability. So really, I think there are a number of innovations that need to occur with mRNA to make them optimally available on a global basis and on a fair allocation system. So these are all parts of things that need to come together over the coming years to prepare indeed for the next epidemic or pandemic.
Thank you very much, Janet and Mel, for your answers there. Any other reporters online who would like to now ask questions of our panel? Yes, James. Feel free to unmute yourself and ask your question and who it's directed to.
Hello, everyone. To be honest, I'm not sure where to direct this question, but I was just wondering if anyone can provide an update on, how much funding has been raised, to date for ACT-Accelerator, and how big the gap is, and what risks, you see at this point, to bringing in the funds that are needed, to fulfill the plans in the coming months. And then if anyone also could comment at all on, how the war in Ukraine is, affecting the broader campaign in terms of trials, deployment, or in any other way. Thanks.
Little bit off our R&D focus here today, James. But as you can see, Dr. Bruce Elwood is online. He was sort of supposed to be listening in, but I think he can probably help with the answer, at least to the first of those questions. So Bruce, I don't know whether you want to come in and help with that.
Yeah, please do so. More interested.
Oh, we can't quite hear you, Bruce. I think you need to put your microphone on.
Okay, can I be heard? Pleased to help,
Good.
but I was here like you, James, to learn from all the other people doing the crucial work on R&D here and support the group. But in terms of the ACT-Accelerator, very quickly. So in the first budget cycle, which ran through the end of 2021, the world was really generous. $18.9 billion was raised toward that work. If we look at now what's needed for the current strategic plan, which runs through September of this year, we need 16.8 billion in urgent, let's say, grant financing, of which we've had 855 million in commitments so far. That does not include the new commitment you heard from Germany just announced 2 days ago, which is fabulous, $1.22 billion. And Charlotte, we can provide the numbers, specifics after, but that's the ballpark. There have also been some new announcements in the run-up to the CEPI replenishment, which is so important next week. That also will overlap with a bit of the ACT-A budget, but we have to see how that plays out, because the CEPI replenishment covers their big agenda, crucial agenda through this next period. So those are what the big picture numbers look like. But the bottom line, James, is super urgent to get the grant financing in place. We've got to get testing way scaled up, as Marta highlighted. Big gap on that right now. Super important agenda to try and get the new therapeutics out to complement the agenda. Like, it's great, the question raised earlier by Anne, I believe it was, but we also need the financing to be able to leverage the work that we're doing with Merck, Pfizer, and others. And then the third piece in the vaccine side, most urgently, the R&D piece that Mel spoke to, and the delivery piece as well, getting the vaccines into arms. So closing the gap, absolutely crucial. Sorry, Charlotte, didn't mean to jump in on that. Oh, and the issue around the Ukraine, super concerning, right? Any conflict, infectious diseases, as we said yesterday, ruthlessly exploit the conditions created by war. And we will see, not just COVID, but other infectious hazards. And it complicates everything from the control of those diseases to saving lives of those who are infected, to managing the international spread, and of course, managing the agenda that we're trying to run, because resources get affected, national attentions get affected, and COVID operates in the COVID response at a political level with the tops of government and the finance ministries. And yes, any major event such as the war we're seeing in the Ukraine will have big implications for our ability to manage this global pandemic and exit it as timely as possible.
Would any of our other speakers like to come in on any of those points before we move on? Just wanted to make sure.
I wanted to come in not specifically on these points, but really on the need for future vaccine research, particularly on the pan-coronavirus vaccines that Mel was also talking about. Now, as you know, there's been quite a lot of research in the past on pan-influenza vaccines, but now I think there's a renewed interest and emphasis on developing a pan-sarbecovirus vaccines, because it's clear that the coronavirus family is potentially one of those that can cause future pandemics, just because of the huge number of coronaviruses that exist in nature and in bats. And we've already had 3 in the last 2 decades. And so it is urgent. So the WHO has been convening scientists to really talk about how one would go about developing a pan-sarbecovirus vaccine. And many of these convenings are done jointly with CEPI. We run regular weekly calls on lab assays and animal models. And then it's also very important, and we've started thinking about how some of these new vaccines would be tested, because clearly it's not feasible now to do the traditional kind of controlled, placebo-controlled randomized clinical trials anymore, because vaccines are widely available and we're scaling those up. And so we've also had recently consultations on developing a framework for the assessment of new vaccines, either known platforms or new platforms, and how we would use immunobridging studies with safety studies in order to advance those, because I think Mel and I both pointed out that we have several vaccine candidates in development, and many of them may have advantages over the existing vaccines. So we do need to take them forward. We cannot stall the development of those vaccines. So we have to be quite imaginative and innovative, and we are working with regulatory agencies as well. We're also revising the target product profile. As you remember, WHO set out way back in March, April of 2020, the target product profile for a COVID vaccine. At that time, we were using influenza as a benchmark, and we said anything more than 50% would be acceptable. But of course, now we know that we could do much better than that. So we're revising the target product profiles, looking at prevention of severe disease, as a primary goal, but also at prevention of infection as a very desirable goal to have. And then also coming up with a framework on how one would assess, because the regulatory agencies and WHO's PQ department would have to put out a clear guideline for developers on what sort of data they will need to submit in order to go for emergency use listing or pre-qualification of the next generation of vaccines. So this is completely new. I don't think we've been in this space before. And so it's also forcing scientists, vaccine developers, regulators to come together and discuss. So I think we've, in the last 2 years, made a lot of advances, not just in developing these new products, but also in the ecosystem and environment of doing clinical trials, large platform trials that have been so successful, the regulatory harmonization, the potential for this type of collaborative registration where you can really speed up licensing of products in countries. Thousands, I think 2,500 registrations have happened just in the last year alone. So there's a lot of learnings from, I think, the R&D in COVID and the kind of global collaboration that we saw, and the focus to speed the development of products for other high priority global, public health diseases. Thanks.
Thanks very much, Soumya. Do we have any more questions from the reporters online? Oh, we've got our expert panel here for you.
Can I hazard a follow-up if other people aren't? I don't want to take somebody else's space, but if
it's brief, yep, go ahead, Anne.
Yeah. Yeah, Mel, I just wanted to ask if you could clarify what you meant about Novavax. The reports are that Novavax has already started to supply to European countries. Has it, in fact, started to supply through COVAX?
Novavax is supplying vaccine doses to COVAX, and it is going through the allocation system that we have. So that is obviously the process in terms of allocation. Doses have not, so it is going through the system as we speak. And as you point out, yes, there are doses going to Europe. There are now doses going to COVAX.
But you have any amounts or have deliveries started? I mean, that it's sort of in concrete terms.
Yeah, as they're allocated, they will become visible, obviously, on the system that we have that shows exactly where things are going. Yeah.
The UNICEF dashboard, I believe, is the system that you can look at publicly and to track those. So that's where all that data is housed. Do we have any final questions?
I just want to let people know that we had this big global forum on R&D last week. This was the third big global forum that really brought together the top scientists from around the world that have been working with the WHO on all the different aspects. As you remember, we had this global research roadmap that was developed way back in February of 2020. And then we've had several streams of work covering all the different aspects of research into this virus. Those recordings are available. So this ran over 2 days, and there are about 12 hours, I think, of recordings with some amazing keynote speakers and also a lot of amazing and really rich presentations and discussions. So for those of you who are more interested in going into details on the what's happening and the latest in R&D, I would suggest you can go to our website, look at the WHO R&D blueprint, and you'll be able to find these recordings, and also you'll find the report on the achievements in R&D over the last year that's been brought together in the report. There's a lot of material there.
Thanks, Soumya. And I've just dropped the link in the chat for those people who might be interested, and we can maybe also share. Actually, in the original invitation that you had to this roundtable, the link to the R&D forum was also there. So you'll be able to find all of the recordings if you'd like to go back and look. Okay, before we close, are there any final questions? Yes, go ahead. And would you mind also saying your publication as well? Thank you.
Hi, I'm Rose Russell from the Evening Standard. This is just a quick follow-up question for Soumya. You mentioned immunobridging. It's not something that I'm familiar with or could explain, but if that's necessary for testing for vaccines, could you explain how that works?
Sure, I can start. I'm sure Mel can also come in. So when we talk about immunobridging, it's essentially using the immunological response to the vaccine as the proof of the efficacy of a vaccine. You know that normally one would go through phase 1, which is mainly looking at the safety. Then you go into phase 2, usually in hundreds of people, where you look at what we call immunogenicity, which means the blood of people who've been vaccinated and then take blood from also the placebo recipients, and you look at the immune response. Immune response varies, depending on which virus or bacteria you're looking at. In the case of SARS-CoV-2, the neutralizing antibody response was found to be broadly correlating with, not 100%, but broadly correlating with the efficacy of the vaccine. And that's something that's relatively easy to track. The WHO also has developed standards so that labs can standardize their neutralizing antibody assays against an international standard and report it in international units. And so immunobridging would mean that you have already data from an existing, let's say the mRNA vaccine of Moderna, and then Moderna is now developing a new vaccine on the same platform where they're using let's say a combination of 2 different spike proteins, and they do a trial, and then they take the antibody levels from that group of people and compare to what they found originally in their first trial, and see whether they're more or less at the same level within a certain limit. And then you would call it as, you would then conclude that the new vaccine was producing the same kind of immune response as the previous vaccine, and therefore should have the same effect when injected into people. And so you need not then go into the large phase 3 with tens of thousands of people, but you would, based on the levels of the antibodies, you would conclude on the efficacy of the vaccine. Now, this becomes a little more complex with the new platforms. So that is why we are coming up with a framework that WHO and regulators can use to assess a brand new vaccine that's coming from a manufacturer who hasn't had a COVID vaccine before. But there's also the T-cell responses. So maybe Mel should come in and explain a bit more. It's more difficult to standardize and compare and we know the T-cell responses, that's the cell-mediated immune response as compared to the antibody response. That seems to be more important to protect against severe disease. So it's very important to measure that and to see that vaccines have both types of responses. Because we've seen that with the waning of antibodies in all of these Omicron vaccine effectiveness studies, that the vaccines did not prevent breakthrough infections, but they were still pretty good, 70 to 80% effective against preventing severe disease. So the immune system is complex and it's hard to measure all the components. But so Mel, I'll hand over to you.
Yes, thanks, Soumya. Yeah, just to build on that, I mean, obviously most of the work is done with antibody. So really bridging between a known vaccine with known efficacy and associated immune response to a newer vaccine. I have to say, then after that, then we need to continue to gather data on the effectiveness of those vaccines if they are license-based or EUL based on immunological parameters. So it doesn't mean to say that you can avoid showing efficacy, but you have confidence that it will be effective. Sumya raises a very good point about the T-cell response. So it is usually much more difficult to do large-scale immunological testing for T-cell responses. But nonetheless, having some evaluation of the T-cell response, in particular, that sometimes the T-cell response may be to more conserved parts of the virus, and so more variant proof as an example there. And also an important factor at preventing severe disease. So it is an area where we need to continue to focus on in an R&D perspective to better understand how well these vaccines will perform in the long term. As Sumya pointed out, as antibody may wane, it does not necessarily mean that protection against severe disease. And if we can find markers that give us confidence, that is an important area of research. Thanks.
Thanks both for that response, and hopefully that's helped answer your question, Ros. I know I've certainly learned a lot that I didn't know about 10 minutes ago. So
yes, thank you.
Right, well, I think that brings us probably to the end. So I just wanted to conclude by saying thank you very much to our panel who joined us today, and thank you very much to the journalists who also joined us. So there's no embargo, so any stories can be written immediately after the briefing closes. If reporters online have any follow-up questions, then please do get in touch with myself or Sophia and put those, and we can make sure that they go to the relevant spokespeople. So thanks very much for joining us all, and we look forward to seeing you at our next roundtable, which is taking place in 2 weeks' time on the 17th of March, and we'll be focusing on the topic of medical oxygen access. Thanks very much, and goodbye.